So we have Harish Vashisth here. So he'll be talking about rare events in RNA conformational dynamics and ligand recognition. So it's over to you, Harish.
Okay, thank you.
Yeah.
Well, thank you everyone for coming to this final talk in the entire meeting. In fact, not only in the session. A few minutes back, Nishant was sort of panicking if I'm going to show up, and I was busy with my weekend job of making breakfast for my family on Saturday morning. So I'm grateful for all of you for coming to the meeting. So today I'm gonna speak a little bit about one of the biomolecules that we haven't heard a lot about, at least in this meeting so far. Particularly, I'll be showing you some of our simulation work related to RNA molecules. So before I begin, I wanna say a few words as to why choose RNA. Why RNA has been so less studied compared to proteins and many other biomolecules, and particularly what some of the exciting questions are in the RNA field. So based on my reading, I think one of the key challenges in studying nucleic acids in general have been a lack of experimental structures, particularly for RNA, because RNA is so different from its other cousin, which is DNA, about which we know very stable structures for a long time. RNA is single-stranded, it could be double-stranded. It's conformationally very flexible, and it presents unique challenges for practitioners of molecular simulations that try to optimize these interatomic potentials, force fields for RNA molecules. So if you follow the field, RNA force fields have been fairly challenging. Lots of modifications have been made to them, way more than I think in proteins, and they continue to be modified even until very recently. So this likely has, I think, demotivated a lot of people from pursuing large-scale simulation studies in RNA. But some new developments in the force fields very recently, I think, where they have been tested on fairly long time scale simulations, as well as different kind of structures, like, RNA can come, as I said, in many flavors structurally. And it's been tested and established that force fields currently, especially the AMBER family of force fields that we have used in our entire work, I think are doing a reasonably good job of capturing this.
Harish, are you changing the slides?
Yeah, that's what I was going
No,
to ask.
I haven't changed any slides yet.
Okay.
No.
All right. Yeah.
Yeah.
Sorry.
Right. Yeah. So it has essentially, the force fields currently, in my view, are reasonably good, and they are being used quite a bit. And I'll show you all of our work I was saying is conducted with the AMBER force field. And for the MD software, our primary tools are NAMD as well as AMBER. No good rationale other than our ability to modify these codes and our experience in working with these codes for a long time. So there's no particular preference other than that. So in probing these questions of RNA conformational dynamics, where should one start? One is always looking for, as we saw in other talks, good model systems that are somehow well characterized, previously studied, where comparisons could be made. One such system that we have started to work on is this small RNA molecule known as transactivation response element RNA, or TAR RNA. It's a fairly small RNA molecule, comes from the HIV-1 virus. So if you look at the genome of the HIV-1, which is roughly about 10,000 nucleotides long, and if you zoom into one end of this genome, I think the 5' end of the genome has this very small hairpin-like structure. This is called the TAR RNA molecule, and I'm showing you its sequence as well as its structures as it is experimentally characterized. So this TAR RNA molecule is known to contain basically two short helices, helix 1 down here and helix 2, which are formed by fairly stable complementary base pairs. And these two helices are connected by this flexible bulge motif shown in red in the structure, and a small loop motif that's at the top. Now, these motifs, loop and bulge, are not just there to provide the flexibility to this RNA, but in physiological sense, and especially how the virus replicates and impacts the cell, has a lot to do with the loop dynamics as well as the bulge. Because if you express the mutants that lack that can disrupt the bulge or loop motifs, you see remarkable change in the ability of the virus to replicate. And some phenomenal experiments have been done on this this specific RNA molecule, and I welcome you to look at, I think, significant work by the Hashim Al-Hashimi's lab in University of North Carolina. And he has applied NMR-based methods to basically probe the dynamics of this molecule in various kind of states. So when we chose this molecule, one of the reasons we made the choice is not only its biological significance and small structure, but more importantly, the conformational diversity that is observed in its structure. In my understanding, I think this is only one of the few RNA molecules that has been shown to bind to so many different kind of molecules. So on this slide, if you're looking on the left, many structures of the TAR RNA are shown. At the center, you are looking at the experimental APO structure, and this is the PDB code for the experimental APO structure. And then on this circular arrangement, you are looking at the same RNA molecule in different conformations, but bound to various kinds of molecules. So you have fairly small molecules on this side, and if you are paying attention, this one, this one, this one, and on this side are very large cyclic peptides that can bind to this RNA. And they cause various degree of conformational change in this RNA molecule. And this is fascinating and useful for us as we try to do simulations with one of these structures or more of these structures, primarily because we know molecular dynamics is very sensitive to initial coordinates. So we want to start with as good coordinates as possible that are experimentally resolved instead of guessing the positions of atoms. So if you map the location of these various ligands, I'm showing you on the right-hand side in a single map, these green spheres are the centers of masses of various kind of small drug-like molecules that have been found to be bound to TAR RNA in experimental structures. And these magenta color spheres represent the peptides that have been found. So most of the small molecules are near the lower helix, shown in cyan, and most of the peptides are near the helix 2, which is shown in blue here. So some of the analysis, you may be wondering the strength of the binding. So dissociation constants for these various kind of ligands are in the micromolar range. Some are weak binders, some are fairly tight binders. The buried surface area between these ligands and the TAR RNA molecule is on the order of, again, 400 to 1700 angstrom square. And if you wonder about how much conformational change is there, then the RMSD is somewhere between 5 to 8 angstroms. So what we did was we did not want to choose one structure over the other, and moreover, we are interested in exploring the general conformational dynamics of this molecule, try to understand how RNA molecules behave in solution. So we started by doing simulations of every single structure that's shown on this slide. We started simulations with the experimental APO structure, and then we created many APO structures by deleting the ligands from them, as well as conducted simulations by keeping ligands present. So we conducted all these simulations and generated, I believe, to date, one of the largest data set on this RNA molecules, somewhere in the range of 60 microseconds of all atom explicit solvent simulations. And these data sets are available to the scientific community at this website. And there is a DOI I can provide later if you need to access the data set for because lots of people we heard are doing machine learning based analysis. We haven't attempted to do any machine learning on any of the conformations we have sampled, but anyone is free to download the data set and try some of their methods. So let me quickly describe what we find by simulating all of these structures of TAR RNA. And I'm going to describe some of the global changes first, followed by local changes and their coupling. So one of the common beliefs that we have studying proteins is that when ligands bind to biomolecules, typically they stabilize the structure. And we wanted to see if that kind of theme holds even for RNA molecules. And indeed, it does to a large extent based on study of this TAR RNA molecule. So if you look at RMSD distribution in the unliganded state and the liganded state, you clearly see liganded structure shifting toward a lower RMSD value, meaning that globally the overall RNA structure got more stabilized in the presence of the ligand. And then we can map these kind of mean values in the liganded state shown here in the light color, and the mean values for the liganded state shown in the darker color for all RNA structures that we simulated. And we find across the board that most of the ligands try to stabilize the RNA structure. So that's kind of consistent with what one would expect, that ligands somehow add to the stability of the RNA molecule, except one of those cases, which is shown here with an asterisk on it, where we see liganded state more flexible than the unliganded state, and I'll show you why that is so. Then we looked into at the next level, so outside of the global RMSD comparison, these two helices that I spoke to you earlier about have been probed and their rotation using NMR methods. And it's known that these helices also become more stable in the presence of ligands. So if you take a vector going along the axis of one helix, another vector, measure the angle between these two vectors, we find that in the unliganded state, these distributions are much more wider and shifted to the right. Meaning that there's more flexibility between the helices. But as you add the ligand, or if you conduct the liganded simulation, you tend to see the distribution become narrower, meaning that fewer values of this angle are sampled, and in fact lower values. So it also kind of rigidifies the helices a bit. So then we next probed what happens at the where is this coming from? So one can analyze residue fluctuations for every nucleotide in every structure. So I'm showing you these maps where x-axis shows individual nucleotides in each of the TAR RNA structure, and y-axis shows a difference in the root mean square fluctuations compared to the unliganded state. So if the ligand stabilize, you are going to see negative values of RMSF, which we see in most of those structures. As you look at this one, this one, this one, this one, this one, this one. But there are a few cases, as I pointed out, in one of those structures where we see positive RMSF values, meaning that a higher or increased flexibility, same in this case. But generally speaking, ligands seem to stabilize not just the helices, but across the board, the entire RNA molecule is stabilized, as indicated by negative RMSF values for all of these structures. One can look at the flipping of bases. So here is one of those bases that in the unliganded state freely flips because it's not stabilized by anything. But in the liganded state, it's significantly stabilized and it does not flip. And we see this kind of stabilization again in many other structures. But let me show you some of the bizarre things we observed. You can call them rarely observed transitions or structures in this TAR RNA. So in one of those structures of TAR RNA, where we start with ligand bound state, we observe that over the course of the simulation, a very large scale conformational change happens. For example, if this was to be considered palm of a hand, then somehow you see the ligand molecule squeezing between it during the simulation. And this is one of those cases where we see different behavior than expected. So even in the presence of the point that I'm trying to get across is that it's not the binding of the ligand itself that stabilizes, but even when ligands are bound, this is an experimental structure, even then we can observe way more conformational changes as we see. This leads to a significant increase in the buried surface area as expected, more atoms are closer. And then this other example is exactly opposite of that. So here ligand was present, RNA was more open, it became more compact. But for a different ligand, we observed that RNA initially was more compact and it became way more elongated. And this has to do with some conformational rearrangements that happened due to insertion and flipping of or rotation of the ligand itself in the binding pocket. So ligand doesn't occupy the state that's precisely in the experimental structure, but it can readjust during the simulation. So these were interesting observations that were different than expected. We also probed if we can observe flipping of bases between the liganded and unliganded state, and somehow they are different. So earlier I showed you that the bases flip less when ligands are bound, but this slide is trying to show you exactly opposite of that. So here is this 4 pair of bases somewhere in the middle of this RNA molecule, U23, U25, and C24. These 3 bases we try to track. And if you look at over the course of simulation, one of the dihedral angles that kind of characterizes whether the base will flip or not. In the initial case, we find that the base is in the outward conformation. So this light gray rectangle is the range of angles within which, if the angle is observed, the base would be considered inward, otherwise it would be considered outward. So initially it was in the outward conformation, but we found irrespective of presence of ligand or not, it could go inward. Similarly, in these 2 other cases, we start with an inward conformation, and we find that irrespective of the unliganded or liganded state, the base can still flip. Similarly, in the 3rd case. So we were intrigued by generally these observations that ligands are somehow able to cause even more flexibility, although generally ligands try to stabilize the RNA molecules. And same for bases, that bases are generally stabilized, but there are cases where even in the presence of ligand, these bases could flip. So we undertook 2 further studies to focus a little bit on applying some methods to understand how these conformational transitions are coupled with ligand binding. One of those studies is shown here. We chose one of these small molecules that can that is somewhat relevant, and its KD is known, it's less toxic. It can be well studied using these pulling simulations. So we carried out constant velocity steered MD simulation. I give some important information on general questions that are being asked on steered MD. It's a fairly old technique, but still very, very relevant and useful method to understand ligand dissociation. And I can go into details of why we choose steered MD over other techniques. So we ran lots of these simulations. You have to be extremely careful to make sure that the ligand fully dissociates and you get force convergence toward the end. And then you can collect the work of dissociation from each of these trajectories and make a distribution. So some of these trajectories show as we dissociate the ligand alone, the same reaction coordinate, we get fairly low work values, but then in other trajectories, we can get high work values. And earlier in one of the talks, I think we heard about the well-known Jarzynski's equality that connects the non-equilibrium work to equilibrium free energy change. And it just so happens the nature of this equality says that we are doing exponential averaging of these work values, which means the values that are in the tails of distributions, especially these lower values, are going to contribute significantly to the overall free energy change. So we can take the work data and convert it into a free energy profile. We try to do it using standard Jarzynski equation and a cumulant expansion of the Jarzynski's equality that's been previously proposed by Schulten and co-workers. And it improves the convergence of the PMF a little bit. You can see in the dissociated state, the free energy is higher. In the associated state, free energy is lower. But we were not so much interested in necessarily obtaining a value for free energy. This free energy value is useful. I mean, we can back calculate the dissociation constant. We can compare with the experimental value, which is anyways known. but we are more interested in as to what happens in these unique trajectories that require lower work to dissociate the ligand. So we further probe these details. I'll point out a couple of unique observations. So the ligand is shown here squeezed between bunch of nucleotides that are surrounding it. And the two nucleotides that I want to draw your attention to is C24, which is right there shown in blue, and U23, which is shown in light orangish or yellowish color. What we observe that in the simulations that lead to a lower work, we see unique arrangements in the conformation of this base C24. So as ligand tries to dissociate out of this binding pocket, initially this base was somewhere outside, a little bit outside the pocket. And as soon as ligand moved out, the base rotated spontaneously inward and occupied the space that was roughly occupied by where the ligand atoms were. On the other hand, in the higher work simulation, this is one of the highest work simulations from the distributions I was showing. So if you take one of these trajectories at the top, we find that there's no such conformational change in C24, although ligand dissociates somewhat in a somewhat similar manner. This contributes to a difference in work values. More importantly, this higher work, I believe, is coming from the orangish nucleotide, where it comes in the way of the dissociating ligand. So there is obviously work required for opening this door or moving the atoms out of way, and that adds to the overall work of the simulation. Then we compare one of the dihedral angles of these bases, and we clearly can capture these transition that in one of the transition is observed in the higher work simulations, SMD dissociation simulations, and the other one is more observed in the lower work simulation. So this was intriguing to us, that there is this coupling of ligand binding, and it could lead to more quasi-equilibrium kind of dissociation of the ligand as opposed to causing significant non-equilibrium as observed in higher work simulations. So you might wonder whether these ideas apply to larger ligands. So I won't go into the detail of this other work, but a recent study that we did has appeared in Biophysical Journal this past month. And we try to apply similar kind of techniques to understand how peptides bind to an RNA molecule. It's an arginine-rich peptide. We study multiple pathways that are shown here. We study the role of restraints. We study dependency on ensembles. Some other reviewer curiosities about whether umbrella sampling will be a good technique, or how can you optimize reaction coordinate using string methods. So all of that is done, and I welcome you to refer to this paper. But let me spend a couple more minutes before I end my talk on another study that we have carried out on understanding the flipping of bases in RNA molecules. So for this particular case, we chose a different RNA molecule that's double-stranded, and where base flipping is more experimentally characterized. So this is a long RNA molecule, longer than the TAR RNA, double-stranded, well-base paired. But here, somewhere in the middle, one of these bases, A18, is known to flip outward. And this transition is basically related to how certain enzymes are going to make modifications on the RNA molecule. So I'm not going into that detail. So the title of the slide says we apply path sampling and likelihood maximization based methods. And we extensively tested many different kind of CVs. We chose dihedral angles that have been in the literature, many distances between bases, across the bases, many angles that we define, number of water molecules, different kind of stacking energies. And we kind of tried all of these parameters to see how well they distinguish between the initial and the final, meaning flipped in versus flipped out states. One of the older parameter has been proposed in an earlier work in 2003. And this is a pseudo dihedral angle that can be defined by pairing this group of atoms and tracking how this base will. So we try to use that older parameter and see if it can well distinguish between initial and final states. And we found, yes, indeed, it does. I show you one of the C trajectories for these calculations, where we observe a sharp transition along this older parameter. We choose some shooting points. We apply these methods by Peter and Trout that was well described, I think, by Srebny Trapter earlier in the meeting. So I'll skip these details. We the key idea in this likelihood maximization method is you can, it allows you to build many models of reaction coordinate by picking single CVs, double CVs, triple CVs, and we did all of these models, roughly 300 of them. And then using a Bayesian information criterion, we can rank them and see which ones are good, which ones are not. And then we eventually come up with our reaction coordinate that turns out to be made up of two unique kind of CVs. One is a distance, stacking distance between the bases, and another is a special type of dihedral angle that captures. So combination of these very well captures the opening of this base. We can project the free energy on our reaction coordinate and look at the transition state, the outward state, the inward state of the base. But more importantly, I want to point out that we discovered this metastable state where we are finding a very unique kind of arrangement of bases. Like in the initial state, only two bases are interacting. In the final state, only two bases are interacting. But in this metastable state, we observe three bases kind of cross interacting with each other. And a key effect of this on the global structure of this RNA is it goes from this magenta conformation to blue. So there is a huge conformational change that happens as these bases rearrange. And this was we were really surprised how the global and local conformational transitions that we observed using path sampling are connected. So with that, I'm going to wrap up my talk and thank two of my students and one postdoc and one student. Dr. Lev Levintov contributed to this work significantly, carried out all studies, and the path sampling work was assisted by Dr. Sanjeev Paul, and he was attending, I think, the meeting earlier. Our studies that I showed you, three kinds of studies. One was on general conformational dynamics of TAR, was published earlier this year. On the small molecule ligand, the steered MD was published this past year, and the more recent peptide one was published last month, and the reaction coordinate one was also published earlier this year. So with that, I want to thank funding agencies that supported this work, NSF, NIH, and the research computing through NSF, as well as University of New Hampshire. I think everyone is ready to brainstorm some, and I'm happy to answer any questions if there are.
So thank you, Harish, for a nice work. So the session is open for discussions now. So we will take up some questions. We have some time for a couple of quick questions. So those would please unmute yourself and you could ask your questions.
Hello, am I audible?
Yeah, maybe you could introduce.
So my question is, you have put this reaction coordinate where you combine the distance and multiply a factor with it and a specific dihedral, right? Hello?
Yes, I can hear you.
Yeah, so my question is, since this distance is a linear variable and the dihedral is a sort of a periodic variable, so how can you combine two different variables of two different nature? For example, if you have a phase shift in the dihedral, then that may change your reaction coordinate.
I get the question. So yeah, you can read the manuscript, I think, and as I said, previous work by Tarapter group, you can normalize the variable and make them dimensionless, I believe. So that's how they are combined.
Yes, Rabani, you want to ask something?
Yes, thank you, Harish, for a very nice talk. I was wondering if you have checked for the kinetics along the reaction coordinate that you have got for your base flipping, and what kind of time scale do you get here?
Right. Well, the thing is, there are no good experimental measurements, first of all, for the system to really know the rate constant. We applied some approximate models. I don't have the numbers with me right now, but they are reported in the thesis of Lev. But yeah, we did some kinetic calculations. That wasn't one of the primary priorities, I think, for us to match the rate constant here. We were more interested in finding a reasonably good reaction coordinate that could capture this transition? And more importantly, can such reaction coordinates, I mean, it's a brainstorming question, can it be translated to other RNA molecules?
Okay, thank you.
Thanks, Ram. So, Peter?
Yeah, hi. Yeah, nice presentation. I was indeed also struck by this same question. So, I mean, the first question that comes to mind here is, so there has been studies in the literature also on DNA and DNA-based flipping. And how do these two processes now compare? So, can you say something about the kind of a general reaction coordinate for these type of biomolecules that do stacking of base pairs?
Right. So yeah, a few different studies, I think, on DNA molecules as early as, I think there was a study in 2003 by Aaron Dinner and Arup Chakraborty. I think very early work on DNA where, well, in their case, I think they create sort of an artificially paired trinucleotide pair, examine the different kind of energies, and they do describe the importance of stacking energies. We did investigate all the stacking energies in our collective variables, and we do not find them. Again, everything is a factor of force field. Force field contributes heavily to the stacking energies, and stacking energies have, for nucleic acids, especially for RNA, have been optimized over a period of time. So I don't know how one makes fair comparison, with previous observation and the newer observations with the newer force fields. But to answer question about general reaction coordinate, I imagine that this reaction coordinate, again, we haven't tested it and it remains to be done. But since it depends on the interbase distance and a particular dihedral angle, these two are very generic parameters that are applicable to any of the RNA molecule that you can imagine. They are not. So we were very curious if water molecules are going to play a huge role here. How stacking energy, and we were somewhat, I think, encouraged that these two kind of variables that we believe, solvent dynamics as well as the stacking energies, could be central, and then it becomes real hard, I think, going from one reaction coordinate to the other, because as the base type changes, the stacking energy changes, and the water molecules are different. So I believe this kind of reaction coordinate, which we explicitly found through this extensive testing, could be useful, I believe, for other RNA molecules.
Okay, thanks very much. Can I actually ask a follow-up question?
Maybe one quick question, Peter.
A quick question. Yeah. So, of course, this is already a lot of work, I understand. But did you also do re-evaluate the quality of the reaction coordinate by an additional committer test and committer histogram of this particular reaction coordinate?
Yes, yeah. I don't have the committer plots here, but I can show you some kind of supplemental data that's in the paper you can look at. So, for example, one can analyze the shooting trajectories that are committing to the reactant and the product states. And you can see if when you project along the reaction coordinate, if you see convergence toward your stable states. And this is just one plot. There is more like this from many other trajectories shown.
Yeah.
If this is what I think the
and these are very expensive, of course. So I
yes, indeed.
Okay. Okay. Thanks very much. I'll
Thank you. Thank you very much. So maybe if there is further questions, you can just write it in the chat here, and or you could write it to Harish directly. So Harish would be willing to answer the questions for you towards you. Yeah. Thank you, Harish. Thank you.